Chapter 18. Blood. In today's lecture, we'll discuss why women tend to have fewer red blood cells than men. It is not because women lose blood during menstruation. That ignores the process of homeostasis. We'll talk about red blood cell production, and right now, I'd like you to take a guess at how many red blood cells you produce every second. And then you can compare that number to the actual number. We will also discuss why red blood cells have this funky shape that's called biconcave. I'll talk about blood doping, which now means two different ways of cheating at sports involving aerobic activity. We'll talk about type O blood being the most common, even though it is recessive to types A and B. And lastly, one of the big topics of today, towards the end, we'll talk about blood clotting. It's absolutely essential to understand the three major steps of blood clotting, and then we'll talk about hemophilia and how it relates, as well as why we might use rat poison in medicine. To do all that, we'll start by talking about the components of blood one at a time, beginning with plasma, and then moving on to the formed elements, including red blood cells, white blood cells, and platelets. Lastly, the big homeostatic mechanism of today's lecture is hemostasis, or blood clotting, which has three major phases: a vascular phase, a platelet phase, and a coagulation phase. Let's begin with blood as a whole. It includes both a fluid component and cells, which are called formed elements. If we were to spin blood down in a centrifuge, about half of it would be plasma, and the other half would be the formed elements. The formed elements include white blood cells, red blood cells, which some people don't consider cells because they lack a nucleus, and platelets, which are really just parts of cells. And for this reason, we lump these three together under the heading of formed elements, cells plus things kind of resembling cells. Let's start with blood plasma, which is the fluid component of blood. It's mostly water, plus all of the dissolved electrolytes and nutrients that we're going to need, and other small molecules, and then a bunch of larger proteins as well. Most of those proteins are made in the liver, and one of the most common ones is albumin. Over half of the proteins in the blood are this one in particular. Albumins help to move steroids and other molecules around the body, things that wouldn't dissolve in water naturally. There's also a number of globulins, including antibodies, which we will discuss next chapter, and then transport proteins like HDLs and LDLs, which will be very important next term. Lastly, fibrinogen is a protein found in the bloodstream, and this will be very important at the end of the lecture today. Moving on to the red blood cells, these make up almost all of the cells found in the bloodstream. I mentioned earlier that some people don't consider these cells because they lack a nucleus. However, they've got the word cell in their name, so I say, who cares? They do last only for about 3 or 4 months. Anytime one of their proteins wears out, it can't be replaced because they have no DNA to transcribe and translate into a new protein. And because they lack mitochondria, they can only do anaerobic respiration. They have this particular shape which is called biconcave, meaning it's concave in this direction and in this direction. This increases the total surface area that a red blood cell has, and that's important for gas exchange, the release of oxygen to our tissues, and the absorption of carbon dioxide. This disc shape also allows red blood cells to form stacks as they enter through small capillaries, and because they're skinnier here in the middle, it even allows them to bend in half, which is important because some capillaries are about half the size of a red blood cell. So this allows the cells to bend in half and then snap back to their original shape much more easily. Assuming, of course, we're not talking about somebody with sickle cell anemia, whose red blood cells are more brittle. Red blood cells contain pretty much just one major protein called hemoglobin. You may have learned in your BI 112 class that hemoglobin has 4 polypeptide subunits, each of which has one heme group. And inside of each heme is one iron atom, and that iron atom is capable of binding a molecule of oxygen. Therefore, one hemoglobin could bind to 4 oxygen molecules total. We lose about 1% of our red blood cells every day, and that equals to 3 million red blood cells produced in the human body in the time it takes me to say 1 alligator. That also means 3 million have just worn out, and it's up to the spleen and the liver to recycle those. I wouldn't want 3 million cells just dying wherever they were sitting at the time inside the human body every second. That could trigger a lot of inflammation. So instead, we recycle those cells. Macrophages are going to help, and those can be found in the liver and the spleen. We can't recycle everything, however. The protein part of hemoglobin can be recycled. It's just broken down into amino acids, and those can be used to make new proteins. The heme, on the other hand, is going to be a little bit more problematic. The iron is recycled, but the rest of that heme group cannot be recycled. The biliverdin, as we will learn next quarter, must be removed from the body by the actions of the liver, the colon, and the circulatory system. For now, though, let's focus on iron. To make all of those red blood cells, we need about 25 milligrams of iron per day, but we can only absorb about 2 milligrams per day from our digestive tract. The other 23 must be recycled. And if the liver is not able to recycle red blood cells properly, we're going to quickly lead to an iron deficiency, which will lead to a deficiency in red blood cell production. One mechanism by which this could occur is if the liver is not capable of recycling red blood cells, and instead they are just dying somewhere in the circulatory system. When this happens, hemoglobin shows up floating around freely in the bloodstream, and eventually the kidneys filter it out and it winds up in the urine where it shouldn't be. Iron deficiencies can also be caused by a lack of iron in the diet or the inability to absorb iron from the diet, such as seen with celiac disease. It can also be caused by an increased loss of iron, especially internal bleeding caused by hookworms and roundworms. On the opposite end of the spectrum is a condition caused by too much iron, or hemochromatosis. This is especially common in Europe, where about 1 in 200 people may have this condition of an iron overload. It's a genetic condition, and the excessive heavy metal in the body can damage some of the other organs, including the kidneys, the pancreas, and the liver. This is probably the only disease that is cured or alleviated by bloodletting, which was the most popular treatment in medicine for thousands of years in Western Europe. When red blood cells are broken down and recycled, the iron was reused, but not the rest of that heme group. That was broken down into biliverdin, which is green, which can be converted into bilirubin, which is yellow. This is done by both the liver and also happens in the intestines. When the liver starts failing, we cannot do this recycling as it should happen. And as red blood cells die, their heme naturally gets converted to biliverdins and bilirubins. If you've ever had a really nasty bruise, you might have noticed it started off black and blue, but as the bruise got older and some of these molecules began to diffuse outwards, you started noticing more of a yellowish and a greenish tint to the borders of that bruise. And that was the gradual breakdown of these molecules on their own. We regulate the number of red blood cells in the body very tightly, and this is monitored by the kidneys, who instruct the bone marrow to make more red blood cells. The stem cells in the bone marrow are called hemocytoblasts. These stem cells, found in the bone marrow, would first undergo mitosis, one of which would remain a stem cell, but the other one could begin the process of differentiating into a new red blood cell. It would start making hemoglobin, kick out all of its organelles to become a reticulocyte, and then it could leave the bone marrow, enter into the bloodstream, and eventually become an erythrocyte, or a mature red blood cell, 1 or 2 days later. This requires all of the raw building materials, including amino acids and iron, as well as a number of vitamins that are not used up in the process. But deficiencies in any one of these B vitamins can lead to a defect in red blood cell production. For instance, pernicious anemia is caused by a lack of vitamin B12. The kidneys regulate how many red blood cells are produced based off of how much oxygen the kidney cells are receiving. If the kidney cells become hypoxic for any reason, for instance, let's say we started bleeding and lost a lot of red blood cells. We now can't send enough oxygen to the kidneys, so they would start making the hormone erythropoietin, which would instruct the bone marrow to make more red blood cells, until finally the oxygen-carrying capacity of the bloodstream could meet the demand of the kidneys. And then the kidneys would stop producing erythropoietin, and red blood cell production would cease. There are a number of things that can decrease oxygen to the kidneys, besides just blood loss. If those red blood cells were defective, or if there was increased oxygen demand, such as moving high in elevation or exercising frequently. All of these things would cause hypoxia of the kidneys and should lead to a compensatory increase in production of red blood cells. We can count the number of red blood cells found in the bloodstream by one of two ways. Machines can give us a red blood cell count, telling us the number of cells in 1 microliter of blood. A little easier to comprehend, though, is a hematocrit, where we take a patient's blood in a capillary tube, spin it down in a centrifuge, and just look at the percentage of what is plasma versus red blood cells. For men, on average, it should be roughly 40 to 55% red blood cells. For women, a little bit lower than that, 35 to 45-ish%. Anemia is a fairly common blood disorder, which is caused by either a lack of red blood cells or defective red blood cells, both of which leads to decreased oxygen levels in the bloodstream. Low red blood cell counts can follow alcoholism, bleeding, certain vitamin deficiencies, whereas defective hemoglobin can arise from an iron deficiency or lead poisoning. On the other end of the spectrum are people who have too many red blood cells. This is much safer than anemia, but it can increase the risk of certain clotting conditions. In sports, there are ways to elevate the number of red blood cells that are considered sportsmanlike, and other ways that are considered cheating. Blood doping can involve the removal of red blood cells from an athlete on one day, and they are saved in the refrigerator. That athlete would then be temporarily anemic, but their kidneys would start producing erythropoietin, and the red bone marrow would quickly replace the red blood cells that had been removed. Then, months later, say on the day of the Tour de France, if you injected the red blood cells that you had saved in the refrigerator, you would now have a larger number of red blood cells in your bloodstream. Hence, you could deliver oxygen to your legs more efficiently and compete in the sport better than your competitors who weren't cheating. Another way of increasing red blood cells that's considered blood doping is simply to inject erythropoietin into the bloodstream. If you use the bioidentical compound, meaning it's the exact same one that your kidneys make, it can be difficult to determine whether you did so by injection or whether your kidneys just make a really large amount of erythropoietin naturally. Either way, to find out if people are blood doping, you need to take multiple tests. You can't just test them on one day. And that's because they have their actual red blood cells and can use their actual erythropoietin hormone. The way to do this in a sportsmanlike way is to train really hard or move to a high elevation, or do both. Both of these things combined lead to hypoxia of kidney cells, which should then produce erythropoietin, increasing red blood cell production until oxygen demand can be met. But then when you move back down to sea level to compete, you will have the advantage over others who haven't been training as hard or haven't been training at altitude. So those are the basic components of blood, including the plasma and the formed elements, including the red blood cells. Next, I'm going to cover blood typing. Red blood cells come in 8 different flavors or types, based on the combinations of proteins on the plasma membrane called the A, the B, and the Rh antigens. Let's start by ignoring Rh and focusing on the ABO blood group. There are 2 surface antigens, the A and the B antigens, and there are 2 antibodies that can stick to those antigens found in the blood plasma, called anti-A and anti-B antibodies. A person's red blood cells can have 0, 1, or both of the surface antigens. Found in their plasma would be the exact opposite antibodies, which means that somebody with type A blood does not have anti-A antibodies. People with no surface antigens would be said to have type O blood. They have neither the A nor the B surface antigen, and they are considered the universal donor. They would have both anti-A and anti-B antibodies in their plasma, which means somebody with type O blood can only receive type O blood. But because there's nothing that an antibody could stick to on the surface of their red blood cells, they can give their red blood cells to anybody else. Somebody with the A antigen inherited the A allele from one of their parents, or possibly both, and they make the A antigen on the surface of their red blood cells. They would be said to have type A blood. They could not and do not have anti-A antibodies in their blood plasma, but they do have the anti-B antibodies. Conversely, somebody who has the B antigen on the surface of their red blood cells would have type B blood and would have the opposite antibodies. They would have anti-A antibodies in their blood plasma. For this reason, somebody with type B blood could receive type B blood or type O blood, but they could not receive red blood cells that had the A antigen on their surface, because their plasma antibodies would attack it. Finally, somebody could have both A and B antigens on the surface of their red blood cells. They would have neither the anti-A nor the anti-B antibodies in their plasma. This person would be said to have type AB blood and would be the universal acceptor. They could receive any blood type because they have no antibodies in their bloodstream that would attack anybody else's blood type. In addition to people having type O, type A, type B, or type AB blood, they also either have or do not have the Rh antigen, which we often abbreviate as negative and positive. People are not born with anti-Rh antibodies, and only Rh negative people can make anti-Rh antibodies, and they only do so after being exposed to Rh positive blood one time. These new anti-Rh antibodies could attack a second improper blood transfusion. So this was different from the ABO blood group, where people were born with the anti-A or anti-B antibodies. Here, we only make anti-Rh after a bad blood transfusion, and only Rh negative people make these antibodies. This, at least, was similar to the previous slide. Nobody ever makes antibodies against their own blood type, and we only make antibodies against antigens on the surface, which means that if you gave Rh negative blood to somebody who is Rh positive, that would be just fine. There's no antigen on the surface of Rh negative for anybody to be making antibodies against. Therefore, we can safely transfuse blood in the following directions. Type O blood can be given to anybody. Type A and types B can also be given to somebody who has type AB blood, but we cannot do these in the reverse because of the antibodies that are not illustrated on this slide. Similarly, we can give Rh negative blood to somebody who is Rh positive. And lastly, we can always give an exact match. Type O can be given to O, A given to A, B given to B, and so on. This makes for 8 different combinations of surface antigens. We have O positive and O negative, A positive, A negative, B positive, B negative, AB positive, and AB negative. Out of all 8 of these, type O positive is the most common. If an improper blood transfusion occurs, the antibodies in the patient's blood will attack the antigens on the surface of the donor's blood, causing a massive transfusion reaction. The injected blood would coagulate and turn into a gel, which cannot be transported through the bloodstream, and this would cause patient death. What must absolutely be avoided is illustrated on the left panel. We cannot give donor cells that have an antigen for which the recipient has antibodies. Those antibodies, because they outnumber the donor cells, would cause all of those cells to coagulate. When we learn about antibody function, this will become more obvious. But for now, if you injected a pint of type A blood into a patient with type O blood, you would wind up with about a pint of coagulated blood. And that's way too much clotted blood in arteries and veins for that patient to survive. On the right-hand panel, we illustrate why, when you can, we like to give an exact match. But if you don't know the patient's blood type, or if you're running low on that specific type, we can always use type O blood as the universal donor. That's because those red blood cells do not contain any antigens that the recipient's antibodies could attack. However, that donated blood does come with some antibodies that could attack the recipient's blood. It's just in this case, the recipient's red blood cells greatly outnumber the antibodies. And so you might get an antibody sticking to a random cell here and there, but that won't be causing any sort of coagulation or transfusion reaction. Nevertheless, we do like to match wherever possible. But in the emergency department, that may not be an option. Here are some numbers that illustrate the percentages of the different blood types in different populations. And depending on where you are looking, the type O blood is actually the most common. In fact, O positive is fairly common amongst many different communities. It's only when you start looking at Native Americans and some others that you don't see nearly as many people with the A and the B mutations. So while O is recessive to the A and B alleles, as you may have learned in your genetics class, the O allele is just much more common, and hence we see a lot more people with type O blood. On the other hand, if you have type AB blood, especially AB negative or any of the other negatives, you probably get called by the Red Cross frequently to donate because of your rare blood type. These days, we rarely see bad blood transfusions, so the only time we're really going to worry about antibodies attacking red blood cells is during pregnancy. If an Rh negative mother has a fetus who has Rh positive blood, the two blood types should not mix because the placenta keeps them separate. However, if anything damages this placenta, blood cells could mix between the two, and during childbirth, the placenta gets damaged entirely. This will allow some of the Rh-positive cells of the fetal bloodstream to enter into mom's circulatory system, and she would start producing anti-Rh antibodies. It takes a couple of weeks to make all of these antibodies, and since childbirth doesn't take 2 weeks to occur, that first Rh-positive child is probably going to be safe. It's the second Rh-positive fetus who needs to worry, because these antibodies, which are much smaller than red blood cells, can get across the placental barrier. And if they do, they could start attacking the Rh-positive blood of that second fetus. For that reason, Rh-negative mothers these days are given drugs like Rogam, which are artificial anti-Rh antibodies. That may seem a little counterintuitive. If one of the fetus's red blood cells gets across the placenta, this drug will destroy it. But that keeps mom's immune system naive. It's like she's never been exposed to Rh-positive red blood cells. And those Rogam antibodies only last for a few weeks anyway, so then they will be gone. And as long as she takes this medication with each pregnancy, she should not develop her own antibodies. And the artificial antibodies of Rogam are made to be too large to ever cross the placenta on their own, hence keeping the Rh-positive fetus safe. So while we definitely want to avoid mixing antibodies with the antigens that they attack inside of a patient, we actually want to do this outside of a patient to identify their blood type. We can isolate these antibodies and use them. If I have a patient who does not know their blood type, I could take a sample of their blood and then dump a bunch of anti-A antibodies onto it. If that patient has the A antigen, those antibodies would cause coagulation, and I would know that they had at least type A blood. I would then repeat the process with anti-B antibodies, and if that did not cause coagulation, I would know they did not have the B antigen. So I would say they have type A, and then I would have to use anti-Rh antibodies to determine if they were positive or negative. And in this photo here, because the anti-Rh antibodies are also causing coagulation, that means the patient does have the Rh antigen, meaning they have A positive blood. So those are the basic blood types, the ABO blood group, in addition to the Rh factor. Next, let's cover the white blood cells. White blood cells contain all of the goodies that a cell should: nucleus, endoplasmic reticulum, mitochondria, etc. The main function of our white blood cells is in the immune system. They fight off foreign invaders, they can remove some of the toxins that they produce, and they can even attack abnormal cells within the human body, such as a dying red blood cell or maybe a cancer cell. Most of our white blood cells exist within the connective tissues of our body, especially lymphatic tissue, but a small number do exist within the bloodstream. For these to be useful, though, they must leave the bloodstream to go attack an infection. And to do so, they must dig their way between endothelial cells to move out of the circulatory system, towards the source of infection. They can detect an infection or damage by the release of inflammatory molecules in that area, and they move towards the inflammatory molecules by a process called chemotaxis. We discussed the mechanism of chemotaxis back in Chapter 14 when I talked about activity-dependent receptor desensitization and activity-dependent receptor internalization. These white blood cells will only detect inflammatory molecules for a few seconds, and then they become desensitized to the signal. And only if they move closer to the signal would they be stimulated again? And this allows them to determine where the infection or trauma has occurred. There are 5 major types of white blood cells: the neutrophils, eosinophils, basophils, monocytes, and lymphocytes. 3 of these have grainy-looking cytoplasms and are called granulocytes. 2 of them have clearer cytoplasms and are called agranulocytes. But that's kind of a goofy way to define these 5 cell types, based off of what they look like. A much more accurate way to lump these into groups would be more developmentally. Let's start with their basic functions, however. Neutrophils are the most predominant white blood cell type, and their major function is to attack bacteria. In fact, they are the first to respond to a bacterial infection. Next up, by numbers, are the lymphocytes. And I'm going to have 2 full lectures on the B cells and the T cells, respectively. Next up, we have a few monocytes, which become macrophages and can gobble up large chunks of debris, such as a dying red blood cell. We have a few eosinophils, and these are useful for attacking parasites. And lastly, basophils, which can release histamines and some other molecules that we frequently associate with allergic reactions. The number of these 5 different types of white blood cells can change depending on the type of infection that we get. For instance, a bacterial infection would see cell division in the bone marrow and production of more neutrophils in the bloodstream. Inflammatory responses often lead to higher levels of the eosinophils and basophils, for instance. Those would be natural increases in white blood cell numbers. Abnormal increases in white blood cell numbers include a number of different cancers, such as leukemia and lymphoma. Patients with elevated white blood cell counts due to these cancers have problems with their immune system, because their white blood cells, despite there being quite a lot of them, are immature and incapable of fighting infections, and therefore people with leukemia are said to be immunodeficient and must be kept safe from the possibility of infection. There are other reasons why people may have too many white blood cells, but more commonly people might have too few white blood cells, or leukopenia, and in this case these patients would also be immunodeficient, not having enough white blood cells to do their job. All of the white blood cells come from this same cell that we learned about 20 minutes ago, the hemocytoblast, which is our stem cell located in the bone marrow. And from this one stem cell, we can either go down the myeloid or the lymphoid pathway to produce the 5 major types of white blood cells. This is the better way of dividing white blood cells into 2 groups. It's either one of these 4, or it's a lymphocyte. This hemocytoblast decides which pathway it's going to go down based off of which hormones it receives. And these hormones are collectively called colony-stimulating factors. There's a whole bunch of different colony-stimulating factors, one for each of these arrows. I am not going to test you on the different colony-stimulating factors, but all of these different ones will drive the production of different types of white blood cells. For instance, MCSF will drive the production of monocytes, whereas GCSF drives the production of one of these granulocytes, not one of these agranulocytes. These colony-stimulating factors can be given to patients to increase the production of their white blood cells if necessary. However, too many white blood cells can be a problem. For instance, mutations to the colony-stimulating factor receptors can lead to a specific tumor type. And if you'd like to know more, you can follow this link here to the journal club that I wrote about the topic. So that's the list of our basic white blood cell types. We need to discuss platelets, though. These are another one of the formed elements, but platelets are not cells. They are chunks of a cell. The actual cell is called a megakaryocyte, and it lives in the bone marrow, and little bits of it break off into the bloodstream to form these platelets. And these platelets are very important in the clotting process. Platelets are only going to last for about a week or so, and after that, we must make more platelets. And as I mentioned, their major function is in the repairing of damage to blood vessels. That leads us to the clotting cascade. This could be a capillary or other blood vessel in cross-section. And at this point in time, the platelets inside of the bloodstream could only come into contact with endothelial cells. However, when there's damage to a blood vessel, things will change. The first phase actually involves smooth muscle. This would be in arteries or veins, not in capillaries, and it's called the vascular phase. Vasoconstriction can occur to try and reduce blood flow and limit blood loss. The next phase is the platelet phase. Platelets can now come into contact with collagen found in the connective tissue superficial to the endothelial cells. And when they do, they become sticky and then attach to more platelets, which will in turn become sticky and attach to more platelets until we form a platelet plug that fills in the gap. This can help reduce blood loss, but it's not very strong. If you've ever suffered a paper cut, the first little red blob that stops blood flow can be easily pushed away and bleeding can resume. So we need something stronger than this platelet plug. And that leads us to the third portion of the clotting cascade, called the coagulation phase. Those platelets release intrinsic factor. Intrinsic meaning the platelets came from inside the blood. And the damaged endothelial cells release extrinsic factor. X meaning these cells are outside of the bloodstream. Only when we get both of these factors released will we start to activate the clotting cascade. There will be a number of enzymes activated, one after the next, which ultimately will lead to the production of fibrin. As this protein becomes insoluble, it traps other cells, like red blood cells and more platelets, forming a scab, or a blood clot. And this will then set the stage to repair the damage. But at least in the meantime, blood loss has been limited. The next couple of slides spell out in detail all of the steps that I covered on the previous one. We start with the vascular phase, which involves the contraction of smooth muscles. Vasoconstriction can narrow the diameter of the damaged artery or vein, but constriction laterally can actually pull these arteries or veins into the skin somewhere. This can also help reduce blood loss, but is very annoying for the surgeon, who now has to go digging around for this blood vessel to close it off, because you, as the surgical technician, did not hand over the hemostats in time. Next up is the platelet phase. Platelets can stick to collagen in a damaged blood vessel, whereas they could not do that in a healthy blood vessel. And this will lead to the formation of the platelet plug. And then we get to the coagulation phase. And we had to have two major signals, the intrinsic and extrinsic pathways. This acts as a fail-safe, so that if an endothelial cell got damaged somehow, but it didn't cause any bleeding, the release of extrinsic factor alone would not be enough to form a blood clot. It would be dangerous to form blood clots in places that we didn't need them. So we wait until we get a backup signal. We also require the release of intrinsic factor. And then both of these will activate a number of enzymes with names like factor 12, factor 11, which activates factor 9, and then we go back and activate factor 10, and as long as we've got extrinsic factor, this will activate the enzyme thrombin. And thrombin can convert the inactive protein fibrinogen into the insoluble protein fibrin, which forms the actual stable blood clot. And here we see the final product. We see a mesh of fibrin proteins trapping a bunch of red blood cells and platelets, and maybe even a white blood cell here, that is sealing up the injury site. Keep in mind that we're trying to create this new structure in the middle of a flowing river. Some of the thrombin that gets activated at the site of injury might get swept downstream. And if nothing were to stop it, it could convert fibrinogen into fibrin and create a blood clot in a location where it's not needed. And this is something the body does not want to have happen. And for that reason, the bloodstream contains a number of anticoagulants. These are proteins or other molecules that block clotting from happening. So the only way for a clot to happen is for the coagulants, such as activated thrombin, to outnumber the anticoagulants. So the first molecules of thrombin that are activated by our coagulation cascade will be inhibited by the anticoagulants. But eventually, thrombin will outnumber the anticoagulants, and we can begin to produce fibrin in this area. And any of those thrombin molecules that get swept downstream, they should be inhibited by the anticoagulants found everywhere. But we're not done yet. We have to make this one step more complicated. And so this extra step is called fibrinolysis. It turns out we're not going to need this blood clot forever. So eventually, we're going to want to destroy it. And it turns out that at the site of injury, we're going to start producing coagulants, like activated thrombin, which can start converting fibrinogen into fibrin, sealing up the injury. But these damaged endothelial cells also produce fibrinolytic molecules, and these slowly build up around the site of injury. These fibrinolytics include things like plasmin. Most of the plasmin is going to quickly be inactivated by antiplasmin, but eventually, plasmin outweighs the antiplasmin. And when this happens, the plasmin can then start dissolving the blood clot. This takes some time, though. We've given the body enough time to heal the injury, and so the fibrin is no longer needed. So it's going to take days or weeks for the plasmin to build up to levels to where we can start destroying the fibrin clot. If you've ever administered TPA, that's a drug that can activate plasmin to try and get rid of blood clots in a patient if you know that they've got blood clots that might damage their body. To summarize, we covered the coagulation cascade, and anything in that cascade is considered a coagulant, including the intrinsic and extrinsic factors, any of those other factors that had Roman numerals, some of the cofactors like calcium and vitamin K, and then the last two, which I am going to require you to know on the test, thrombin and fibrin. The activity of these coagulants can be inhibited downstream by anticoagulants, such as heparin. You might have administered heparin to a patient to try and prevent them from forming blood clots. For instance, if you've had to remove their blood for dialysis, you don't want that blood clotting when it's inside the dialysis machine. Lastly, when the blood clot is no longer needed, a fibrinolytic can destroy the blood clot, and that includes the enzyme plasmin and a molecule that activates it called TPA. Sometimes the anticoagulants, or the fibrinolytics, don't do their job as well as they should, and a blood clot forms in a place where it's not needed. This can restrict blood flow in that area. A thrombus is a blood clot that forms in a fixed location, and this can lead to high blood pressure. An embolus is a blood clot that is formed but then dislodges and begins floating freely in the bloodstream. Eventually, it'll wind up in a small enough blood vessel, causing a blockage elsewhere. If this is in the brain, we would call that a stroke or a cerebral vascular accident. So both a thrombus and an embolus are definitely things we want to prevent in our patients. Here are 3 common chemicals that are used to prevent the formation of a thrombus or embolus. Aspirin can block the activity of platelets, and therefore blocks the second phase of hemostasis. Heparin, on the other hand, was one of those natural anticoagulants that we learned about, and can block the coagulation phase. Warfarin also blocks the coagulation phase. High enough levels of warfarin means that the blood becomes too thin, and internal bleeding leads to death. That is why warfarin, at high doses, can also be used as a rat poison. But at low doses, we can just inhibit enough clotting to prevent the formation of a thrombus or embolus, not so much that it leads to internal bleeding. If you're curious, warfarin has nothing to do with warfare, but has to do with Wisconsin. And it blocks vitamin K, which is a required cofactor for many of the steps in the coagulation cascade. If you ever needed to memorize which steps, there is a little mnemonic here for you to help you remember that it's factor 7 and 10 and let's see, I can't remember what the rest of these stand for. I haven't had to test anybody on this for a long time. Leeches produce a very powerful anticoagulant called Hirudin. This is one of the most powerful anticoagulants that we know, and it also blocks thrombin. Unlike heparin, this is not a molecule that you would find naturally in the human body, but it's pretty important for these leeches, which are trying to live off of your blood, to keep the blood flowing. They don't want clots to form. It happens that this chemical also helps to aid in healing of surgical scars, so they might be used in medicine in different places. Chelation therapy is one of the more dangerous treatments that is used by some alternative medicine practitioners, and they really shouldn't be doing it. A chelator is a chemical that can bind to a heavy metal, and then this helps to pull the heavy metals out of the bloodstream and excreted into the urine. Heavy metals that might be found in the human body include lead, calcium, arsenic, and iron. This is why we don't frequently use chelation therapy in medicine. If somebody had lead poisoning, you could use a chelator to get the lead out of their bloodstream. Unfortunately, it'll also get rid of the iron from their bloodstream and calcium as well. An iron deficiency can lead to anemia, especially if chelation therapy is repeated frequently, and a calcium deficiency leads to an inability to form blood clots, which can lead to internal bleeding and death. And that's because calcium was a necessary cofactor for many of the enzymes in the coagulation cascade. But definitely, chelation therapy is not a cure for any of these diseases. And not only does it not work in the treatment of these diseases, it's dangerous because of its effects on iron and calcium levels in the body. It's true that iron and calcium would be very dangerous to the human body if the human body didn't have ways of safely storing them, like in hemoglobin for iron and in bone tissue for calcium. Next, I'm going to discuss some diseases that affect the coagulation cascade, and the first are hemophilias. There are a number of different types of hemophilia, but these are inherited diseases where people are born with 2 mutated copies of one of the factors found in the coagulation cascade. If you don't have any factor 8, or if you don't have any factor 9, or if you don't have any factor 11, it means that the coagulation cascade can't finish. It doesn't matter where you stop in the middle, you don't get to the production of fibrin, and this means blood clots can't form, and people are at a greatly increased risk of bleeding. For instance, this gentleman over here has some pretty serious bruising, And this was probably not caused by massive trauma, but just by sitting and standing and walking around, bumping into people at the grocery store. That causes small damage to our capillaries, which gets healed very quickly, thanks to a healthy coagulation cascade. But without that, you can see the effects here. Serious internal bleeding from what the rest of us would consider not even a minor injury, but something we never noticed at all. There are other effects of hemophilia besides the inability to form blood clots. It also causes damage to joints. Our treatments for hemophilia currently are not very good. Blood transfusions are fairly common, but that gets to be expensive over time, and especially during the 1980s, when people couldn't be sure whether their blood being transfused was free of the HIV virus. This was pretty dangerous. The downstream factors can also be injected into the patient, meaning one of those factors with the Roman numerals after it. We try and activate the pathway later if one of the earlier steps is blocked. Lastly, there's been some advances in making GMO human beings, using a virus to inject the healthy gene into these patients. And so far in preliminary trials, the results have been amazing. It's actually cured the hemophilia. But this is still in trial and is not being used in medicine currently. Another bleeding disorder is caused by a defect in platelets, and this is called thrombocytopenia. It has the same result, which is the clotting cannot occur properly, which can lead to internal bleeding. It's just the pattern of that bleeding looks different in a patient with abnormal platelets than it does in a patient who has abnormal clotting factors. So we've covered the function of platelets now, and they were involved in blood clotting, or hemostasis. Our first phase of the hemostasis was a vascular phase, meaning muscles constricted to reduce blood loss. And then our second phase was the platelet phase, where the platelets stuck to collagen. And those platelets plus the damaged endothelial cells would then trigger the coagulation cascade, where a bunch of clotting factors in the bloodstream ultimately led to the activation and formation of fibrin. We then moved on to discuss blood clots in the wrong places, namely a thrombus and an embolus, and how we could prevent or even get rid of a thrombus or an embolus with the use of an anticoagulant. And lastly, we covered a few diseases that were caused by defects in the coagulation cascade. But that should wrap us up for this chapter.